I'm Nikita. I am from the Department of Chemistry in the University of Massachusetts, and I'm working with Lynn Marie Thompson. I'm a postdoc in her lab, and we use solid-state NMR to study membrane proteins with a focus on protein dynamics, as we know that dynamics is important to understand the function and sometimes the mechanism of action for many proteins. So that's the thing that we focus on. So today I'll be talking about protein dynamics that we experience in our system, that's the bacterial chemoreceptors, and the challenges that came with it, but also the unexpected ways that it helped us to study the protein in detail. So briefly, what is our system? We work with bacterial chemoreceptors, especially the TAR or the aspartate receptors. These are really long proteins, which are homodimers. These homodimers are form trimers of dimers, and they are associated with two other proteins, the key A, which is a histidine kinase, and the key W, which is a coupling protein, and they form these beautiful hexagonal arrays. Now, together they form these mega Dalton assemblies, and so you can guess why solid-state NMR is needed to actually study them. But what is important to see here is they form really beautiful base plates at the cytoplasmic end of the bacteria. And with such large base plates holding the trimers together, one might expect that the assembly might be fairly rigid, but we were surprised by the extent of dynamics that we actually encountered in this complex. So why do we believe that dynamics is important for this system? So to answer that, we need to have a look at how these receptors work. So upon ligand binding, there is a 2 angstrom piston motion in the membrane region and just below the membrane region that is called the HAMP domain. And then somehow magically the system, the signal is propagated through this long 200 angstrom tip to the histidine kinase. Now, depending on whether the ligand is bound or unbound, the kinase switches on or off. And if it's on, the bacteria starts to tumble towards its food source. Now, this particular reaction is also coupled with an important reaction, which is the methylation of the receptors. So we have made 2 mutants in our lab, the CF4E, that is the cytoplasmic fragment with 4E. That is, the methylation sites are mutated to glutamate residues, so that forms our unmethylated receptor. And for CF4Q, where all the methylation sites here are mutated to glutamine residues, which mimic the methylation. So since this region, the mechanism of this region is already known, our lab focuses on just the cytoplasmic fragment of this protein. And the 2 major questions that we are looking at is whether there is a change in dynamics if the kinase is on or off, or the receptor is methylated or unmethylated, and is there a change in the receptor contacts that it makes with the kinase as well as the coupling protein as the signal switches. So just to reiterate, when the kinase is off, the methylation is on, and when the kinase is on, the methylation is off. Before going to the data, let me just show briefly how we actually prepare the samples. So we do have functionalized lipid membranes. So they have NTA chemistry, and our labeled protein, that is the cytoplasmic domain, is purified with histidine tag. So classic his NTA chemistry gives us these beautiful arrays. And when we add the key A, that is the histidine kinase, and the coupling protein, they form native-like arrays. So this is our pellet that we actually put in the NMR rotor. And as you can see, the stoichiometry of the array is intact in our sample. They are active and they form native-like arrays. So all good to go. The first question that we now want to ask is there a difference in dynamics between the methylated and the unmethylated state of the receptor? So the very first experiments that people do in solid state NMR is to first see what kind of a signal we are getting. So the cross-polarization experiments, basically we can observe the rigid segments of the protein, and with inept experiments, we can observe the flexible segments of the protein. And as you can see here, we have much higher inept intensity in the unmethylated state than the methylated state. And the CP signal is higher for the methylated state. So this definitely tells us that the unmethylated receptor exhibits a greater dynamics than the methylated state of the receptor. So now, of course, with a 1D experiment, you cannot identify what are the residues involved in this segment. So we ended up doing a 2-dimensional inept experiment. Now, if you see here, the first thing that we observe is, as expected, the red signal that is coming from the unmethylated state is of higher intensity than the methylated state, but you can see that the resolution is not sufficient to identify the individual residues. But we were able to identify the amino acid type in this spectrum. Now, a previous grad student in the lab had worked on the CF4Q sample, and they had identified that in CF4Q, the N-terminus methylation helix 1, as well as the C-terminal tail is dynamic. She quantified the inept intensities coming from the spectrum to actually identify these regions. I will not be talking much about it, but I'll be happy to share how we did that later in the talk, after the talk, or maybe in the breakout rooms. So the two important things for us to note here are that all the peaks, whether it's from CF4E or from CF4Q, are at random coil chemical shifts, and there are no new peaks in the CF4E spectrum. So this indicates that there is no major conformational change occurring between the two receptors. Now, coming back to the increased intensity, it is possible that the increase in intensity in inept fraction may be a result of the change in proton T2 between the two samples. So that's what we checked. So once we calculated the proton T2 of the inept fraction for the methylated and unmethylated receptor, we realized that they're not too different. So they don't really account for the twice an increase in the intensity that we are looking at. So we closely analyzed the 2D experimental data that we had for both the receptors, and we realized that the increase in inept intensity was not uniform throughout the spectrum. So there were certain peaks that were showing much higher change in intensity than the others. So we used this data to actually understand which amino acid types were specifically increased in the CF4E spectrum. And this indicates that probably there is an additional dynamic segment in CF4E. So what we did was we redid the peak volume analysis similar to what we had done in the CF4Q sample. And we realized that upon checking the sequence, we realized that another segment of the protein, that is towards the C-terminus and the MH2, methylation helix 2 in CF4E, was also dynamic in the unmethylated state. So just to reiterate what we saw was there was an increase in dynamics when the receptor switches from kinase on to kinase off. So definitely our unmethylated receptor has higher dynamics. And one thing to note here is the increase in dynamics is at the where the methylation sites are. So this might increase the access of these sites to the methyltransferase enzymes during the signaling. So once we kind of got the dynamics part sorted, we shifted our focus to the rest of the receptor, that is where we think which should be rigid. So we already know that around 30% of the residues in it are inept visible in our receptors. So we anticipated that we should be able to probe the rest of the 70%. So the rest, all the green region, we should be able to see. However, our initial experiments with CP-based, initial CP-based experiments did not really give us a really good signal. So when we tried to check the efficiency of the CP signal, we were getting really low signal, and we checked it against a frozen sample where where there is no dynamics. And we realized that we only have 40% of the signal, whereas we were expecting around 70% of the signal if we just remove the inept visible part. So this could mean two things. Either the green region is CP visible, but the entire green region is CP visible, but it has a low CP efficiency, or only some part of this green region is visible here in this unfrozen spectrum, and all of that has a high CP efficiency. So just to see which one of the two we are seeing, we probe the CP buildup curves, and we see that the unfrozen buildup curves for both CF4E and CF4Q matches the frozen CP buildup curves if we normalize it for the intensity differences. So this actually means that with the rise being same for the unfrozen and frozen, it would indicate that only 40% of the sample is actually CP visible, and the rest of the protein has intermediate dynamics, and probably we will not be able to probe it either by CP or inept-based experiments. So the number of CP observable residues that we might actually have is maybe around 100 residues in both the mutants. So in one way, it is a challenge that we are not able to see significant part of our receptor, but in the other way, it is actually an opportunity to understand much more about the CP observable region, and we believe that this would give us better peaks and a better resolution because we have a really long receptor. So just to see if we are indeed getting it, we started probing we did some double CP experiments to probe where these rigid regions are coming from. Now we have some amazing spectra that we got from this protein, but we still haven't assigned all the peaks. But another grad student in the lab had done some HTX-MS studies on the sample, and her results showed that there was a slower D2O uptake in the protein interaction region. So right at the tip of the receptor, it showed slower deuterium uptake. So we believe that probably the peaks that we are seeing here might include the PIR region. So is there a difference in this rigid region when the kinase is switched on or off? Definitely, when we look at the spectrum, we see that there is a huge chemical shift. There are huge chemical shift changes that are happening between the two samples. So there might be a difference in the way the receptor is actually contacting the kinase or KW, or there are some other major structural conformational changes that are occurring between the two mutants. So we are really excited to understand which one of the two it might be. But that's when we hit another challenge. And we think that's also because of the high dynamics in the sample. So we realized that our samples are not staying stable. So when we make our arrays in solution, they remain active for almost 2 weeks. But when we pellet it down for NMR studies, our pellets were losing activity within 3 days. So, and this also corresponds with how the sample in the NMR was behaving. We were seeing a reduction in CP intensity, whereas an increase in the inept intensity. So the sample was progressively getting destabilized probably. So maybe there is a loss in native structure, or maybe the sample is shifting from kinase on to kinase off states. But the bigger challenge is that we cannot really do longer NMR experiments if we want to figure out the sequence specific resonance assignments. That's when we used another strategy to make our samples, and we made samples with PEG mediated assembly. So here, instead of the lipid membranes, we actually used PEG as a crowding agent to make our samples. And these samples also form these beautiful native-like arrays. So we are not really distorting the native structure, hopefully, for the protein. Now, these samples are actually working out really great for us. As you can see, the activity, even after 7 days, we still retain 90% of the activity of the receptor. The CP signal is very stable, and there is just a minor fluctuation in inept signal, and we don't think it should matter too much to us when we do longer experiments. So we are already in the process of working out this sample, and we have got, like, this is fresh out of the oven data. We have already started some experiments with it, and it is looking really good for us. One of the important things that we are really excited about is, for the first time, we are able to see some glycine peaks in the NCA spectrum here. And why I am so excited about these glycine peaks is when we look at the sequence of the receptor, we'll see that the glycines are actually occur either in the protein interaction region or in the middle of the flexible bundle region. So one thing is, this entire region corresponds to around 100 residues, which exactly matches the calculation that we just did 2 slides back. And another important thing is that this glycine hinge region is where a lot of cryo-EM studies have shown that the receptor starts splaying from this particular region. So the very fact that we are seeing these peaks in our spectrum is showing us that probably not only our calculations are right, but our hypothesis with the rigid region being in the protein interaction region might also be right. So this is, of course, not complete yet, but it does give us an excellent opportunity to finally identify what is going on in the regions which are not so dynamic, and how the contacts with the kinase and the coupling proteins might be actually changing. So I'll just summarize real quick. Hopefully I've shown you that these chemoreceptors are really exciting systems which show both considerable dynamic and considerable rigidity, and everything in the middle, which we still have to figure out. Between the 2 states of the receptor, the unmethylated state of the receptor shows higher dynamics, and there are definitely structural changes occurring here, which we are trying to figure out what. Finally, I would like to acknowledge all the members of the Thompson Lab, especially Lynn Marie for her mentorship, and Katie here helped me do some of the experiments. The 2 previous grad students, Mariam, who did some experiments with 4Q, and Shuni, who did some HDXMS studies. All the experiments were carried out in the NMR core user facility at UMass, with Yasna and Vego helping us with it, and the project was funded with NIH.